which through the help of different chemokines and cytokines, then infiltrate into the tumor microenvironment. Depending on the cell types and the resident immune cells present in the tumor microenvironment, if the cytotoxic T lymphocytes can initiate clearing of tumor cells via inducing cell death by different mechanisms like apoptosis, autophagy, and necrosis. The presence of immune evasive cells like MSDCs, cancer-associated fibroblasts, and tumor-associated macrophages can inhibit the T-cell mediated cytotoxic response in the tumor cells. So, to identify the exact molecular mechanisms and the type of TLR receptor expression is very crucial to generate an effective anti-tumor response, because TLR stimulation or tumor cells, the type of TLR that is expressed and activated on tumor cell can either lead to tumor progression, metastasis, recurrence, and chemoresistance. Also, activation of certain TLRs, which I will talk about in the later part of the talk, can also induce cell death in the tumor cells. Similarly, the type of TLR that is expressed on immune cell type, like if TLR is activated on Treg, can promote immune suppression, or if the TLR is primed and activated on dendritic cells or NK cells, it can promote activation of T-cells, which can help in tumor clearance. Plus, TLR expression also activated on other cell types such as endothelial cells and fibroblasts can both ways activate angiogenesis or anti-angiogenesis or anti-metastatic response, depending on the cytokine and chemokine molecules that are present in the tumor vicinity, and also on the type of other immune cells that are present in the tumor microenvironment.
She has not yet been added. SDG and IC.
Okay. So, to further assess the scenario, toll-like receptor also play a significant role.
She has not yet been added. SDG and IC.
Different cell types present in the tumor microenvironment.
Is trying hard to fight back. We tried our best to present today's discussion on health science and human diseases.
Glycolytic pathways and lactate
For
acid
those
production, upregulation of
who is just now joining,
also upregulate
a very warm welcome
which leads to activation
and a little
of
mention
cells,
for the whole audience.
and in macrophages,
The link
upregulation
for
of
feedback
TLR2 and TLR4
form,
has
the link for
shown
feedback
the
form will be
glycolytic
given on the
pathways
chat
and
box
increase
towards the
CA
end of the
and
second
oxidative
question answer
phosphorylation
session,
pathways.
and will be
Similarly,
there
in cancer
for a little while after the webinar is over as well.
Has
Please feel free
induced
to
pro-tumorigenic
respond with your
response, while
convenience.
activation of certain TLRs like TLR7 and 8 has generated anti-tumor effect when combination when administered in combination with different metabolic inhibitors.
Due to some urgency, our principal sir has not able been to join us right now. Instead,
Thus,
we are fortunate
these data show
to have
that
him
targeting
on a record.
the metabolic
Let's hear him
pathway
online.
is also in the
For
immune
this,
cells and the
I'll
cancer
call
cells
upon
can help
Srimati Bonani
improve the
Bindani
TLR
to connect.
targeted therapy
Over to you,
in cancer
Srimati
patients.
Bindani.
Thus, the role of TLRs
Here
is
you are.
quite
Yes,
important
yes,
for
sir.
generating both innate and adaptive immune responses in cancer patients. Shown here is an example of how TLR agonist has been used as adjuvant in cancer vaccine trial, where TLR agonists in response to different PAM signal lead to priming and activation of help in priming and activation of naive T cells into effective T cells, which then help to initiate an effective anti-tumor response in the patient. Thus, innate immune factors like toll-like receptors could be effective immune targets for enhancing anti-tumor response, and potential TLR agonists in regulating the response to chemotherapy is currently being evaluated in cancer patients in different trials. Now, the next question that comes to mind is, how is the toll-like receptor expression altered in different cancers? Now, there are several molecular mechanisms that have been reported that can significantly alter the TLR expression and TLR-mediated signaling in cancer. Shown here is the table which shows the different genetic polymorphisms that have been reported in different toll-like receptor genes. The polymorphisms in TLR2, TLR4, TLR5, and TLR9 have been associated with enhanced susceptibility to different cancers, including breast cancer, melanoma, hepatocellular cancer, prostate cancer, and bladder cancer. In fact, another polymorphism in TLR2 promoter has been seen in multiple cancer to be associated with enhanced risk of cancer. Similarly, polymorphisms in TLR6 gene has been shown to mediate a protective effect against increased risk of breast cancer and colorectal cancer. That is why the status, understanding the status of different polymorphisms in the TLR gene could act as a prognostic marker for predicting the clinical outcome in the cancer patients. Also, the TLR signaling and TLR mediated downstream effectors are also affected by epigenetic regulation, epigenetic modifications. Shown here is one study by Banerjee et al., where stimulating the TLR signaling by different TLR ligands like LPS for TLR4, poly-IC for TLR3, and CpG-ODN for TLR9 caused an increase in ERK MAP kinase signaling, phosphorylation at the IL-10 promoter, which is one of the transcriptional target of TLR pathway activation. Shown here in this figure is an assay where the stimulation of the cells of tumor-associated macrophages and naive macrophages by different TLR agonists led to an increase in histone H3 phosphorylation. Inhibition of the ERK MAP kinase pathway, shown here, PD98059 is an inhibitor of ERK MAP kinase pathway, inhibited the phosphorylation and of the histone H3 at IL-10 promoter. This modification, or the phosphorylation of this histone protein at the IL-10 promoter is associated with upregulation of IL-10 and TGF-beta in the tumor-associated macrophages, which are shown in black, compared to shown here is an increase in IL-10 expression both at mRNA and protein level compared to naive macrophages when stimulated with these ligands. So these data show that epigenetic deregulations can alter and hamper the TLR-mediated signaling in the immune cells. Next, also promoter methylation of the TLR genes also plays an important regulatory role in regulating the TLR expression in both cancer cells and in immune cells. Shown here is one study by Kim et al., where they studied the methylation status at the SP1 binding site. SP1 is a transcription factor that facilitates transcription of several genes. They found in the gastric tumor, compared to normal tissue, the tumors were hypomethylated at TLR4 gene promoter. While in a panel of gastric cancer cell lines, these authors found variable methylation status, where some of the gastric cancer cells showed hypermethylation in the TLR4 gene. When treating these cells with a demethylating agent, which is like azacitidine D, which is a DNMT, the DNA methylase inhibitor. induced hypomethylation in the TLR4 gene promoter. This shows that the TLR promoter methylation could be a frequent event in gastric cancer. A similar correlation with the methylation status and expression of TLR4 was also observed in this. Methylating with azacitidine caused an increase in TLR4 expression in the cells which were earlier hypermethylated. So this shows that expression of the TLR4 protein is correlated with its promoter methylation status. Thus, epigenetic modifications play a crucial role in regulating the TLR gene signaling and TLR gene expression status in different cancers. Now, microRNAs have recently also been reported to play a role in regulating the TLR expression. Shown here is one study by Chen et al., where the different microRNAs like microRNA 21 and 29A were packed in microvesicles were released from the tumor cells as exosomes, which when taken up by led to activation of toll-like receptor 8, which led to secretion of pro-inflammatory cytokine, thereby preventing tumor progression and metastasis. So this is the study in details by this group, where they found microRNA 21 and 29A to be upregulated in lung cancer cell lines. These are the two lung cancer cell lines that they studied. They found, in this figure, they found a co-localization of TLR8 expression with these sets of microRNA, which was further confirmed by a co-immunoprecipitation assays. They found microRNA 21 and 29 to be selectively associated with the TLR8 transmembrane protein in the lysosomes. A similar result was also observed in the mouse lung tumor. My apologies, this is the best figure that was published in this paper. The red color shows the expression of exosomes. CD9 is a marker for the exosomes. The cancer cells, the tumor cells, release the exosome containing the microRNA 29A, which is stained in blue in this section. The tumor, the merged image shows that the endosomes, the microRNA 29A was localized where the microvesicles were present in the normal lung tissue and tumor cell periphery. This shows that the microRNAs via secretion through microvesicles also play an important role in regulating the TLR-mediated responses in different cancer cells. Now, toll-like receptors are expressed not only by cancer cells or immune cells, they are expressed by several different organs in the body, though the highest expression as expected is observed in the immune cells like granulocytes. This is the example of TLR4, but a similar pattern of expression is also seen for other TLRs expressed in humans. Here, TLR4 shows highest expression in granulocytes, monocytes, dendritic cells, and spleen and lymph nodes, but a low to moderate level of expression were also observed in several other tissues. Thus, which suggests that different roles of TLR-mediated activation occurs in different tissue cell types. In addition, this data shows the variable level of TLR expression in several different cancers originating from different organs. This study shows the TLR expression status in gastric cancer tumors. Several studies I have not shown here have correlated the TLR expression with overall survival and other clinical-pathological parameters and even chemotherapy response of different cancer patients. Shown here in this study is that the gastric cancer patients overexpressing TLR5 had a significantly better survival than patients having low expression for TLR5. Thus, it suggests that evaluating the TLR expression could have a prognostic relevance for cancer patients. Now I want to talk about the relevance of toll-like receptors in breast cancer. Pardon me. In my previous research in my postdoctoral lab, we used to study a variable level of cisplatin responsiveness in different panel of triple negative breast cancer cell line. Now, this is the worst subtype of breast cancer with only conventional chemotherapy, radiotherapy, and surgery being the only approved treatment option for these patients. Targeted therapy are not successful for treating these patients because they lack expression for estrogen receptor, progesterone receptor, and HER2 receptor. That is why chemotherapy, which includes paclitaxel, cisplatin, and cyclophosphamide, are the only available treatment approaches for these patients. So we found a variable expression chemotherapy response in these cell lines. Then targeting with combining with small molecule inhibitor, targeting tyrosine kinase c-MET with a small molecule inhibitor in combination with dasatinib, which is also quite frequently administered to these patients, greatly reduce the chemoresistance seen with dasatinib in these breast cancer cell lines. Now, why am I talking about the study in this figure in this talk? It's because it shows that the chemotherapeutic drugs that are available for treating patients has a very variable risk. Thus, targeting, there is a need to target other novel targets which can augment the chemotherapy response in these cancer patients. Now, in my current lab, a study by Dr. Karuna Madan, who heads our department, she found that treatment of the prostate cancer cells with a soluble pattern recognition molecule called surfactant protein D, it reduced the cell viability and induced apoptosis in prostate cancer cells and also human prostate cancer cells derived from cultured from tumor biopsies. And this effect, cytotoxic effect, was selective for prostate cancer cell, causing no such effect in normal prostate epithelial cells. Thus, it seems that targeting the immune response could be a better therapeutic approach for metastatic cancer cells, including breast cancer. That is what promoted me to study the effectiveness of toll-like receptor and agonist in breast cancer, particularly in the subtype triple negative breast cancer, which is one of my favorite research topic. We first did RNA-seq. We first analyzed the TLR expression status in the RNA sequence database submitted in the TCGA datasets. We first segregated the TNBC tumor or the patients into distinct TNBC subtypes. A lot of recent current literature have further subtyped breast cancer into further molecular classifications as shown here. Why is that important? Because these molecular subtypes have their unique enriched signaling profiles, which which has been reported to be responsible for a variable, which has been suggested to be responsible for the variable chemotherapy response seen in this subtype of breast cancer patients. So these subtypes, out of these subtypes, the basal-like 1 and the basal-like 2 subtypes shown here in green color and pink color or purple color show the best paclitaxel therapy, probably because they have mutations in DNA damage repair pathway, and the worst prognosis is seen in LAR subtype, which is the most chemo resistant type. overall prognosis has also been seen in the immunomodulatory subtype, which is shown here in orange. In this subtype, the high throughput sequence analysis has shown that most of the immune signaling pathway are enriched in this particular subtype, and there is a higher percentage of immune cell infiltration also seen in this subtype of triple negative breast cancer patients, which suggests, and recent studies particularly in breast cancer, also in other cancer like lung cancer, it has been seen that infiltration with more number of immune cells, more the number of infiltrating tumor cells, more has been the better prognosis of the cancer patients. Like there has been better survival and better chemotherapy response seen in patients who have more infiltrating lymphocyte populations in their tumors. So we found that most of the TLRs in the patients in the TCGA data sets, the most of the TLRs were overexpressed in this immunomodulatory type. Also, what was interesting was we found TLR3 and TLR4 to be significantly downregulated, not only in the subtypes of TNBC, but also downregulated in other subtypes of breast cancer, that is the hormone receptor positive subtype of breast cancer, shown here in red and blue color bars in the data. So it suggests, this data perhaps suggests us that these TLRs play a very important role in regulating the immune response and also the pathogenesis of triple negative breast tumors. Also, what was interesting is that we found TLR8 to be significantly upregulated, TLR7 and TLR8 and TLR9 to be upregulated in the immunomodulatory subtype compared to its level in normal tissues and also other subtypes of breast cancer. Then we looked at the expression status of these TLRs in different subtypes in different panel of TNBCs. Each of these cell lines that we selected represents unique molecular subtype of TNBC. The first cell line represents the basal-like subtype, the second the basal-like 2 subtype. The third cell line represents immunomodulatory subtype, and this MM231 represents the mesenchymal stem cell subtype, which has enriched stem cell renewal pathway and is more invasive in nature. We found TLR4 to be significantly downregulated compared to normal cell. For us, our normal cell was MCF10A, which is a non-malignant immortalized breast epithelial cell line. Compared to the expression status in MCF10A, TLR4 was downregulated in many of these cell lines. Interestingly, in HCC1187, which is an immune-enriched subtype, most of the TLR expression, though the expression level is low, even in the TCGA data set, the overall transcript level expression of TLRs is astoundingly quite low, up to 1 to 5 transcript per million also for certain TLRs. But however, compared to the normal cell, we found a significant difference in the TLR expression in the immune-enriched subtype, which suggests that TNBC is an immunogenic tumor, and perhaps TLR agonist treatment and in combination or with other immunotherapeutic drugs or with chemotherapeutic drugs could perhaps improve the treatment outcome for this subtype of cancer patients. Next, we also checked the expression profile. Again, we found, as expected, variable levels of expression with high expression in HCC1187, which was an immune-enriched subtype, compared to MCF10, which is a normal or control epithelial cell. We, on treating one of these cell lines with paclitaxel, which is a routine, which is one of the routine chemotherapeutic drugs given to administered to TNBC patients, we found a downregulation in TLR4 mediated signaling, which suggests that exposure to chemotherapeutic drugs alters the TLR mediated signaling in the cancer cell, which could be playing an important role in regulating the anti-tumor immune response. Well, we are planning a lot more studies, but I just wanted to share this part of the data with you all. Another study published way back in 2010, also reported overexpression of TLR3 in tumor cells in breast cancer and TLR9 overexpression in breast cancer, and correlated with the prognosis of breast cancer patient. Here also, the breast cancer patients who overexpressed TLR3 in the breast tumor cells had worse prognosis compared to patients who had low TLR expression in the tumor cells. And in the breast cancer patients where TLR9 was overexpressed in the fibroblasts had a better survival, a significantly better survival than patients with low TLR9 expressing fibroblasts, which suggests some prognostic relevance of these genes in breast cancer. Thus, it seems the accumulating data by different investigators just confuses the scenario much more, where both anti-tumorigenic effect and pro-tumorigenic effect has been reported for different TLR genes. Like shown in this figure, upregulation of TLR2 signaling in cancer cells, I'm not talking about immune cells, in cancer cells has been associated with the pro-tumorigenic response, including inflammation, increased vascularization in the tumor, enhanced metastasis, and immune suppressive microenvironment reported for the cells in the cancer cells. Then similarly, upregulation in TLR3 signaling has also led to reduced apoptosis. Pardon me. Has generated anti-tumorigenic response by inducing apoptosis, necrosis, decreased proliferation, and prolonged survival of cancer patients. Then upregulation of TLR4 in breast cancer, colon cancer, gastric cancer, and lung cancer has been associated with pro-tumorigenic response, again associated with increased viability, enhanced proliferation, tumor growth of the enhanced tumor growth, initiation of carcinogenesis, and induction of immunosuppressive microenvironment. TLR5 in breast cancer, head and neck, and colon cancer has been associated with decreased proliferation of cancer cell, enhanced neutrophil infiltration, whereas upregulation of TLR5 in gastric cancer has been associated with enhanced proliferation of cancer cells. So, according to the microenvironment, the type of exposure to the invasive pathogens, the damage-associated molecular patterns, and the surrounding cell types in the tumor microenvironment of the cells actually regulates the TLR-mediated immune response in different cancers. Similar pro-tumorigenic and anti-tumorigenic responses has also been reported for TLR7, TLR8, and TLR9 agonists. Several other studies have also tried to correlate the different TLR expression with prognosis of different cancer. So like the previous slide, some TLRs like TLR4, TLR7, and TLR9 has been correlated with poor prognosis, as indicated in this study, and some TLRs like TLR4, 3, 4, and 9 has been associated with good prognosis in hepatocellular cancer, breast cancer, and renal cancer patients. Recent studies have also suggested a prognostic role for soluble toll-like receptors. Soluble toll-like receptors have been detected in saliva, plasma, amniotic fluid, and in human breast milk. They are secreted in the extracellular environment through cleavage via matrix metalloproteases like ADAM10, ADAM17, which releases the which cleaves the extracellular fragment of the TLR from the extracellular fragment. This extracellular fragment has the capacity to bind with different TLR agonists and also with the extracellular fragment of other TLR membrane receptors expressed by immune cells and cancer cells and other cell types. By binding to the extracellular domain of with other TLR membrane TLR receptors, these extracellular domain act like a feedback inhibitor of the TLR signaling itself. The molecular mechanisms, till now, soluble ectodomain shedding of TLR2, TLR4 and TLR9 has been reported so far in both normal human in different fluids and also in different cancers. The molecular mechanisms identified so far has been alternative. Hello. So far for this process mechanism is alternative corresponding mRNAs coding the TLR genes and also proteolytic cleavage of the TLR protein. This is an example where the feedback mechanism used by soluble TLR receptors has been shown to evade has been shown to evade the pro-inflammatory response. Now, this is in intestinal epithelium cells in absence of soluble TLR2 receptors. In presence of invading pathogens, the membrane TLR2 is activated, which leads to upregulation in TLR-mediated signaling, thereby causing an increase in pro-inflammatory cytokine and chemokine secretion, which leads to infiltration by several immune cell types. Now, in case of certain pathogenic infections like HIV, which can itself invade and proliferate further in the immune cells. The infiltration by these immune cells is an invitation for further propagation of these pathogens. In presence of soluble TLR2 receptors, the membrane TLR2 mediated infiltration of the innate immune cell response is inhibited, which is which has a positive role to play in inhibiting the pathogen-mediated immune response. But in cancer scenario, what is the role of soluble TLR receptor is still not known, but different investigators are still evaluating the prognostic relevance of soluble TLR receptors in response to different chemotherapy and immunotherapeutic treatment. So this is one study where increase in soluble TLR expression was seen in progressive stages of lung cancer, and the prognostic also patients with high soluble TLR4 expression had a better survival compared to patients with low soluble TLR level. In another study conducted at Banaras Hindu University, also reported reduced level of serum TLR9 in patients of benign breast diseases and breast cancer patients. So, measuring the soluble TLR receptor could play an important role in prognosis of cancer. So, the next question that comes is, can targeting toll-like receptor signaling improve the prognosis and treatment of cancer patients? The doctor here seems to be very positive about the role of TLR as a cancer chemotherapy agent. Now, the table here shows the different studies that have reported both pro-tumorigenic and anti-tumorigenic response of different TLRs. In one study in breast mice model, combination of TLR7 with cyclophosphamide and radiotherapy regressed the tumor response, while a TLR4 upregulation in cancer cell has been associated with increased paclitaxel resistance. Now, in another study by Willens et al. On melanoma mice tumor model, the group of mice treated with TLR7 agonist MED19197 significantly improved the survival of mice compared to mice which were treated with vehicle or with Resequimod, which is another commonly used TLR8 agonists. Similarly, in breast tumor model and colon cancer model, the mice treated with TLR8 agonist significantly reduced the tumor burden. Also, treatment of the melanoma in melanoma model, treatment with the TLR agonist with OXO4 antibody targeting OXO4, which is a tumor-specific antigen, greatly reduced the tumor volume compared to TLR8 when TLR8 was treated alone or in combination, or when the antibody was treated as a monotherapeutic agent. Now, these are some of the TLR agonists that are commonly being evaluated in different clinical trials targeting different TLRs. In recent studies, several studies are evaluating the potential of TLR agonist as an adjuvant in different cancer vaccine therapies to modulate the CD4 positive T cell response. Now, the table here shows the different current ongoing cancer vaccine trials where TLRs have been used as adjuvant in vaccine therapies treating breast cancer patients. Some of these studies are still ongoing, and in one of these studies, TLR3 has been used in combination with immunotherapeutic drugs or monoclonal antibodies targeting HER2 breast cancer. Now, these are some other studies where other TLR agonist has been used in combination with both immunotherapeutic drugs and chemotherapeutic drugs. In one phase 1 study which has been completed, they found that when TLR7 agonist imiquimod was administered in combination with cyclophosphamide and radiotherapy, 2 out of the 10 patients showed partial response to this combination of treatment, and changes in TIL, infiltrating TIL profiler was also seen, where there was increase in CD4 positive T cell was seen in the tumor tissues, and local production of like IL-6 and IL-10 decreased in this patient with increase in interferon gamma seen in one of the responders. So the TLR agonist, either as monotherapy or in combination therapy, still seems promising to some extent. These are some of the approved TLR agonists. BCG, which is a TLR 2 and 4 agonist, has been approved for treating bladder cancer. MPL, which is a lipophosphosaccharides LPS derivative, is also been approved as an adjuvant for cereberic, which is given as a prophylactic treatment to prevent cervical cancer. And imiquimod, which targets TLR7 signaling, has been approved for treating basal cell carcinoma patients. Now, these are some other trials which are either ongoing, targeting different TLR agonists and several different cancers. Now, why is it that we still do not have approved TLR agonists for treating different cancer patients? These are examples of some failed trials, like the trial with TLR3 agonist was administered in combination with poly-IC and immunotherapeutic drugs was soon withdrawn. Also, in this another trial with TLR8 agonist with cyclophosphamide was terminated after enrolling just 4 patients. Most of the clinical trials with toll-like receptor as monotherapeutic agents have failed because TLR agonists can also activate the immune suppressive pathway as a part of feedback mechanism via induction of immune suppressive cytokines like IL-10. When TLR signaling is upregulated in Treg cell, it can generate an immune suppressive response. Also, upregulation of PD-L1 is often seen along with stimulation of TLR agonists. So, a number of novel strategies are now needed to overcome the immune suppression which is induced by TLR stimulation in different cancer. One approach could be using combinatorial therapeutic approach, where the TLR agonist, combination of TLR agonists can be tested with chemotherapeutic drugs or radiotherapy treatment, or combining the TLR agonists with different immune checkpoint blockers could help improve the therapeutic efficacy of TLR agonists. Also, stratifying the patient population according to the TLR expression status can help generate better response to TLR ligand as vaccines or as monotherapeutic agents. Also, identifying novel molecular markers to predict the response to these cancer vaccines or TLR ligand therapy can also improve the therapeutic efficacy of the already used TLR ligands. Also, there is a need to find novel TLR agonists. New stimulatory elements could improve the therapeutic response induced by the TLR agonists. A novel TLR agonist with potent immunostimulating capacity will certainly advance the field of TLR agonists in terms of cancer therapy as cancer immunotherapeutic strategy. Now, future perspectives. Using TLR agonist as adjuvant in cancer vaccine is already but there is a need to identify more effective TLR agonists can be included as adjuvant in vaccine formulations. Also, tests can be developed that can directly assess the potential of different TLR agonists to activate innate immune and adaptive immune response. This can be done by checking the expression profile of different immune suppressor protein, evaluate the tumor infiltrating lymphocyte status in the tumor, which can give idea which patient is going to respond better. Like shown here in this figure, patient A might have a mutation A coding for a particular immune receptor protein, and patient B can have a mutation 2 coding for a different kind of a protein. So maybe a TLR agonist A could be more effective for treating patient A, while TLR agonist B could be more effective for treating patient B and not for patient A. Also, understanding in details the molecular pathways which are getting affected by TLR stimulation or which are regulating the TLR agonist stimulated signaling pathway are also needed to fully understand and improve the effectiveness of TLR agonists as chemotherapeutic agents. Most of the TLR agonists till date have failed as monotherapy agents due to self-regulated immune suppression and IL-10 upregulation. So, identifying novel therapeutic candidates to overcome tolerance of the tumor cell to TLR agonists is warranted. Example, IL-10 blockade improves the imiquimod-mediated anti-tumor efficacy and survival in vivo model of several cancers. Another approach could be using nanoparticles for efficient delivery of TLR agonists like R848, which is a TLR7 and 8 agonist. When loaded with cyclodextrin nanoparticle, led to efficient drug delivery to tumor-associated macrophages in vivo. Also, use of using acid pH responsive PLGA lysosome loaded with TLR7 and 8 agonist elicited a stronger antigen-specific CD8 T cell response and NK cell response against tumor cells, as shown in this figure. This is another study where nanoparticles, acid pH sensitive nanoparticles loaded with TLR7 and 8 agonist augmented the NK cell mediated antibody-based cancer immunotherapy response. Here, the nanoparticles in response to changing pH inside in the dendritic cell led to secretion of pro-inflammatory cytokines and and augmented NK cell activity. Now, this study here has used a nanoparticle formulation enriched with containing TLR7 and 8 agonist using two model of cancer. The first model is a model of breast cancer, where the mice were administered with an antibody targeting EGFR, because EGFR is often upregulated in breast cancer cells, along with followed by subsequent treatment with nanoparticles loaded with TLR agonists. Now, administrating the nanoparticle loaded TLR7 and 8 agonist induced higher infiltration of NK cells in the tumor, as shown in this figure, and also lesser tumor burden was observed when the mice were treated with this combination of TLR8 agonist with the monoclonal antibody against EGFR. Also, significantly better survival was seen in the mice when they were treated with this nanoparticles loaded TLR8 with the antibody against EGFR. Thus, this could be a very effective strategy for exposing cancer cells to different TLR agonists. In another tumor model, which is a lung tumor model, when the mice were treated with cetuximab and TLR8 loaded nanoparticles, more infiltration of NK cell was observed in the mice administered with this formulation. Thus, to summarize the results, ongoing clinical trials involving a series of TLR agonists support the notion that such agonists can be safe and effective immunotherapeutic candidates as adjuvants. Besides, combination therapy with TLR agonists could improve the therapeutic efficacy and clinical outcome of cancer patients. Thus, to sum up, the role of TLR signaling and TLR agonists seem to be promising as a therapeutic strategy in cancer patients. However, more efficient markers and therapeutic strategies are needed to overcome the resistance that are eventually developed while treating with such agonists. With that, I end my talk. Just want to introduce our Innate Immunity Lab in our institute. She's Dr. Taruna Madan. She's scientist F and she heads our department. Her research interests include evaluation of innate immune molecules as microcytes for prevention of HIV, role of collectins in uterine biology, pregnancy maintenance, and immunoprivilege of testes. And she's also studying the role of recombinant human SPD, which is another soluble pattern recognition receptor. She's studying its role in pathogenesis of prostate cancer. And that's me. My research interests include molecular genetics and molecular immunobiology of breast and prostate cancer. And I am also interested in understanding the role of stem cell renewal pathway in pathogenesis of reproductive cancer and other reproductive disorders. So this is our institute. This is how it looks like from our gate. And I just wanted to share our institute has been privileged with a high throughput COVID-19 testing facility. It was inaugurated just 3 days back on 27 July. Our institute is one of the 3 institutes in India where this high throughput testing facility has been established. Our Honorable Prime Minister Narendra Modi inaugurated it virtually. She is Dr. Smita Mahale, our institute director, and he's a senior scientist who is a part of COVID team. COVID-19 team. Our institute is also a center for COVID-19 testing facility, and where the validation and confirmation of COVID-19 testing is done in Mumbai region. So now for the acknowledgements, I want to thank my institute director, Dr. Mahale, for all the support I have received, and hopefully will receive while I'm here, and our department head, Dr. Karuna Madan. She's a great mentor, and she has been with me for initiating my research activities in the institute. And Dr. Deepak Modi is a renowned embryologist and a very good friend and mentor for me. We very often have deep scientific discussions. And I also want to thank Mayur, who joined one of my breast cancer projects recently. He works very hard to take the project further. I also want to thank Dr. Somnath Mukhopadhyay and Dr. Bonani, who invited me for this lecture. Hopefully it has been informative for you all. I also want to thank the funding agency who helped me run the lab-related activities on a daily basis. And lastly, I want to thank the patient audience who has been patient and kind enough to give their time to hear me out. So a big thank you. And yeah.
Thank you, Dr. Mukherjee. Thanks for your such an enlightening talk. Now, the topic is open for interaction. While all questions, it may not be possible to address all the questions for paucity of time. So, I request now Srimati Bonani Bindhani, Assistant Professor, Department of Zoology, Dinabandhu Andrews College, to kindly conduct the question and answer session. Over to you.
Polymorphism is concerned, not side effects have not been reported. The only impact of these polymorphism is an upregulation in TLR signaling or their association with increased chemotherapy response. But whether there is any cytotoxicity related with the polymorphisms, that I won't be able to say. BCG has different domains. See, the LRR domain, as I have shown in the figure, in the toll-like receptor, has a binding affinity for different TLRs. There are different TLR signalings which have binding specificities with distinct ligands. So it turns out that the BCG has a binding affinity or a signaling sequence that can bind with the LRR domains of TLR2 and TLR4 receptors. I hope that helps you to some extent.
Thank you, Professor Bindhani. First of all, I would like to congratulate and thank Madam Speaker for such an enlightening and informative talk. Thank you, Madam, once again. One other question which I saw from the participants is that, can you, ma'am, please enlighten us on the mode of action of coronavirus targeting apoptotic signaling pathway in research? Are there any roles in anti-apoptotic and apoptotic genes?
See, I will not be the right person to discuss that because I don't work with coronaviruses. So my apologies on not being able to answer that question. I would not really like to answer that question just out of the blue.
Okay, ma'am, thank you. And the last question is, can you briefly state the status of TLRS mediated treatment on different types of cancers in India?
In India, to my best of my knowledge, no clinical trials have been initiated. However, in my project, I am collaborating with Tata Medical Center, and hopefully, our study on Indian cancer patients will throw some light on the TLR agonists on treating at least breast cancer patients. So, hopefully, our study is going to be the first in this scenario.
Thank you, ma'am. Thank you very much on behalf of the entire department and entire college of our college. Thank you, ma'am. Thank you so much. Now I request Srimati Ruksa Noor, Assistant Professor, Department of Zoology, Dinabandhu Andrews College, to kindly introduce our second speaker, Dr. Nandan Kumar Mondol. Over to Srimati Ruksa Noor.
Thank you so much, ma'am. It's my great privilege to introduce Dr. Nandan Kumar Mondal. He is an experienced biomedical researcher. He completed his both graduation and post-graduation degree from the University of Burdwan and was the recipient of University Academic Gold Medal as a first-class first student. He completed his MPhil in cancer cytogenetics in the year 2007 from the University of Burdwan. He completed his PhD degree from CNCI in 2011 and has joined as assistant professor at Department of Zoology, Kavi Nazrul College of the University of Burdwan. In 2014, he joined as postdoctoral fellow at University of Maryland School of Medicine. He became acquainted not only with human subject research, but also with GLP standard animal studies. In 2015, Dr. Mondal joined University of Louisville in the Department of Cardiovascular and Thoracic Surgery. And in 2018, Dr. Mondal joined as assistant professor at Michael DeBakey Department of Surgery, Cardiothoracic Transplantation and Circulatory Support, Baylor College of Medicine, Houston, Texas, United States, to continue his research on heart failure, heart transplantation, circulatory support. And to his credit, he has published more than 50 peer-reviewed national and international research articles. His long-term goal is to advance the understanding of biomedical devices induce blood cell dysfunction and to use this knowledge to guide the clinical practice and the design improvements of biomedical devices. Sir, we are very privileged to have you here. Over to you, sir. Kindly. Over to you, sir.
Hello, can you hear me?
Yes, sir, you are audible, sir.
Am I visible? Can you see me? Because I don't know whether you can see me or not.
Yes, sir. You are.
Yes, sir. Yes, sir. Yes, sir.
Okay, got it. Thank you for the nice interactions. So it's an honor to be a part of this one day international webinar on health science and human diseases organized by the Dinabandhu Andrews College. And it's my pleasure to be a part and to present my few of ideas and few of understanding about the heart failure and heart transplantations. And I would like to I would like to thank you to the principal of the Andrews College, Dr. Mukhopadhyay, and also Srimati Bandhani for inviting me for this webinar. And now, can you see my slide? Can you see my slide? Okay, so I'm ready to present. Can you see this? So can you see my slides here? I just
yes, absolutely, sir.
Yes, sir.
Okay.
Yes, sir. You're
Okay,
clear.
got you. So we're ready to go. So,
You're
okay,
clear.
got you. So we're ready to go. So today I'm going to discuss about the current scenario and future perspectives of heart failure, biomedical devices, and heart transplantations. So before I go into the deep of my presentations, I'd like to say a few words. Most of you already know that what is human heart, how it works, what are the heart diseases, what is heart failure, and what are the treatments for heart failure. So in the slides, you can see the human body and its total circulatory systems and how the heart is connected to the lungs and throughout the body, and how the oxygen is delivered to the blood, and then less oxygenated blood come to the heart for oxygenation and goes to the lungs for oxygenation. So these are the basic things. I'm not going to elaborate this because you are all college students or university students, so everybody knows about the human physiology, how the heart, the central pumping organ works at its base. So as you can see, the heart is covered with the sac that's called the pericardium, and in between the heart and the pericardium, there's a pericardial fluid. It's an insulator that protects the heart from mechanical shock or damages. So human heart and circulations, again, this is the flow chart, and everybody knows from the very beginning that how the circulations occurs to the heart. So I'm not going to elaborate this. And there is another video. I don't know whether you can see it or not, because the media is not found. Okay, no matter. Now I'm going to tell you about what are the types of the heart disease. So the congestive heart cardiomyopathy and valvular heart disease are among the heart disease types. There are more than 50 types, each affecting different areas of the heart and or the blood vessel systems within it. Some people are born with certain types of heart disease known as the congenital heart disease, and other develops over time. So what is heart failure? So heart failure is a chronic progressive condition in which the heart muscles is unable to pump enough blood through it to meet its body's normal demand for oxygen. Basically, the heart cannot keep up its workflow. So there is a picture. It's the picture of the failing heart, and this is the normal heart. As you can see, the weakened heart muscles cannot pump when it's enlarged. So if we look at the classification of the heart failure, I took this paper from the one of the Indian journals. So they nicely classified according to the criteria of the heart failure. So on the basis of the left ventricular ejection fractions, the heart failure they classified. On the basis of the time course, they classified the heart failure, and on the basis of the severity of symptoms. So this is the classifications in United States. So we generally follow the class 1, 2, 3, and 4. And I'm not going to elaborate on what are these class and how these are happening. So in these slides, I'm going to show you a little bit of video animation to see how a healthy heart, heart failure, congestive heart failure occurs. Generally, a heart can beat approximately 60 to 100 times a minute, and again, I don't know, this video is not working. We had a nice animations of congestive heart failure, how it occurs. So unfortunately, I don't know, it's not playing. So I am going to skip these slides and go to the next section. So this is the cardiovascular disease in the United States. So cardiovascular disease is the leading cause of mortality globally. The heart failure alone is 5.8 million patients in the United States, and 1 in 5 people dying within 1 year, and fewer than 60% surviving 5 years. So the total cost of the heart cardiovascular disease in the United States is 444 billion. Now I'm looking to the heart failure scenario in India. So I just searched the web and found the Indian Journal of Dental, Indian Heart Journals, and found that, and also compared this data to this report, American Heart Association's report. As you can see, most of the places are blank, the white, the no data. So as there is a loss of data, we cannot come with a proper statistics the current scenario of the heart failure in India. But the heart failure is associated with the shorter life expectancy, increased frequency of the hypertension, and the poor quality of life, and major public health challenges in India. Treatment of heart failures. So medicines, as you all know, that if you have a little bit of heart problems, there are several medications that the doctor prescribes to correct your problems. But this is not the current topic of my presentation. So the medications is the common medication that is used to treat the heart disease or failure. So what are the actual treatment for the heart failure is the surgery or medical device, because heart failure means your heart cannot pump enough oxygen or enough blood. So the heart is not working properly. So either we have to replace this heart or we have to correct. So we need a corrective surgery. We need a supportive surgery so that the heart can pump enough blood to the rest of the body. Or ultimately, if the heart is not at all working, we need to replace the heart. So the surgery is coronary bypass surgery, valve repair or replacement surgery, implantable cardiovascular defibrillator, cardiac resynchronization therapy, biventricular pacemakers. These are all different kinds of surgeries, and ultimately the heart pump. When the heart cannot pump enough blood, we will support the heart with a mechanical pump made up of metal, and that can assist the heart to make their function properly, to get the blood from the ventricle and then deliver the oxygen and nutrients throughout the body by facilitating the proper beating of the heart. So this is the heart pump that can facilitate the heart, that can be supported a damaged heart to function properly, or until the patient got transplanted or the patient has a donor heart for transplantation. So the ultimate solution for the heart failure is the heart transplantations. But it's not that easy. If you need a heart, it's not like that. I got admitted to the hospitals and the doctor diagnosed me the heart failure. Okay, so next step you need a heart, and they can order the heart, and tomorrow you'll get the transplant. No, this is not the way. This is what. So we have to wait. There are a lot of people waiting for the heart transplantation. So you have to be in the waiting list, and you have to wait. How long? That depends on at which number you're in the waiting list. And there has to be the heart has to be matched with you. So during that time, if your heart is not properly working, so what you should do, we will use a support device that is called the ventricular assist device to support your heart until you got the heart transplantations. So what is the ventricular assist device? So ventricular assist device is a mechanical pump that is used to support heart function and blood flow in people who have weakened hearts. So this device gets the blood from here and then circulates, then goes back to the aorta and circulate to the bed, throughout the body. So actually it can facilitate the working of the ventricles. So that can support during surgery until heart recovers, while waiting for the heart transplantation, and if not eligible, so that can be used for long-term durations. So what are the basic part of the ventricular assist device? Again, I have a nice video here to show how this works, but it's not working. So you can get an idea from this picture. So in this picture, you can see this is a pad made up with metal, and it's a big because it's an older device. It's a pulsatile device used in long back ago. So this figure, you can see the blood comes here. Here there is a motor and blood goes back to the so that it facilitates that heart functions, and that is connected to the outside power source outside the body, and so this is the basic part. If you saw the video, then it would be nice, but it cannot be. So now I am going to a little bit about types of VAD. So VAD can be connected. VAD can support a left ventricle. VAD can support a right ventricle. VAD can support both. So two basic types of VADs are the left ventricular assist device that support left ventricle, and the right ventricular assist device that can support right ventricle. If both types are used at the same time, they are called the biventricular assist device. So this is the left ventricular assist device. Can you see? Oh, this is the left ventricular assist device, the most common type of VAD, and it helps the left ventricle pump blood to the aorta. The aorta is the main artery that carries oxygen-rich blood from the heart to the body. Now, the right ventricular assist device usually are used only for the short-term support of the right ventricle after LVAD surgery or heart surgery. And our VAD helps the right ventricle pump blood to the pulmonary artery. This is the artery that carries blood from the heart to the lungs to pick up oxygen. And this is the biventricular, both. When both by LVAD and RVAD are used together to support. This is called the biventricular assist device. Now I'm going to give an overview of the VAD, the basic designs. So there are two types of design. One is transcutaneous VAD, and another. I'm coming to the next slide. So the transcutaneous VAD has its pump and power source located outside the body. So pump and power source located outside of the body, and the tubes connects to the heart through a small hole in the abdomen, and this type of VAD might be used for short-term support during the surgery. And another is implantable VAD. Implantable VAD means it has to be implanted inside your chest. So you have to make an incision and put this VAD inside your body, and it connects to the ventricles. It takes blood from here, it pumps here, and then goes back to the aorta. So this kind of implantable VAD had its pump located inside your body and its power source located outside the body. So far I am telling about the VAD. So as you see in this picture, the VAD has many generations. So VAD is not a new thing, but every day we are getting a newer technology. So from older technology to newer technology, as you can see, the first generation VAD was a pulse style VAD. That is the heart made, and it's a very big. And now we improve in size, we reduces the size, we increase their functionality, we make them more biocompatible through the technology advancement. So older technology to newer technology, now we have a third generation of continuous flow centrifugal pumps, that is the hardware HVAD and the HeartMate 3. We do have HeartMate 2 also. So this is second generation. Now we have the third generations in HeartMate 3 and the hardware HVAD. So now I'm going to tell you about the history of innovation, the Texas Heart Institute, the Baylor College of Medicine. This is the institution where I currently work. So why I am interested to tell about the history of innovation, because the Texas Heart Institute is established in 1962, and the first successful heart transplant 1968, first artificial heart implantation 1969 in the United States done in here, and then the cardiovascular surgery research laboratory established in 1972. Then again, the second total artificial heart implanted in 1981 here in our institutions. Then first rotary blood pump implanted in 1988 by Dr. Fraser. And then we have the first pre established the implantation in the United States at our institution, the 2003, and this is the total artificial heart as we see the design. So these are the different part of the total artificial heart that can replace the total healthy human heart with this device. So a lot of research is going on with this. Total artificial heart is here. So here I have a video. This video is working. So now we can have an animation. So this kind of device, that is the rotary total artificial heart, is the powerful device that can be connected to the chest cavity instead of heart transplantations. And this artificial machine can facilitate can act as a heart, not as a biological heart, but as a machine heart. And I'm not going to discuss the artificial heart in these presentations because my focus is on heart transplant and the VAD because limited time. So maybe in the later next time I will discuss a little bit more about this artificial heart. I just wanted to show how it looked like and how. So now coming to the division of the cardiothoracic transplantation and circulatory support. This is our divisions of the Baylor College of Medicine Department of Surgery. So I work in these divisions. And the development of the total artificial heart was initiated by the Michael E. DeBakey in the Baylor lab in 1950s. Dr. DeBakey obtained a first federal grant in 1960 to develop artificial heart. And Baylor has remained world leader in the surgical treatment of heart failure. So this is Dr. Denton A. Cooley. So the first successful cardiac transplantation in the United States was performed at Baylor St. Louis Memorial Center by Dr. Denton A. Cooley, 1968. So we are the first by Dr. Denton A. Cooley. Dr. Michael E. DeBakey Department of Surgery was established. Now we do have the Dr. Noon, Dr. Bud Fraser, and Dr. William Cohn. These three people are currently still active in our department, and I'm very fortunate to work with them. And under the leadership of Dr. Bud Fraser, a total of more than 1,300 heart transplants and over 1,200 VAD implants have been performed at our institutions. So he is the living leader. So he developed the HeartMate 1, HeartMate 2, HeartMate 3, all this kind of device he developed, and he is the pioneer, and he is the living legends of our institution, still working. He's now 80 years old, still working, and still come every morning and actively involved with all kind of research activities. In 1963, first successful pneumatic LVAD implant in the US at our institution. This is Dr. DeBakey, Dr. Cooley, Dr. Fraser. They're all working together. And these are the different types of the VAD. This is the older generation. Now different types of the HeartMate 3, the HVAD, the HeartMate 2, the Jarvik, the all other pumps. So we are the first, because I am telling you that the Texas Heart Institution and Baylor College of Medicine, these are the world famous, the number one institutions in terms of the device implant, device design, device innovations, heart transplantations. So 1970, the first bridge to transplant LVAD in US. Bridge to transplant, BTT means bridge to transplant means a patient is waiting to get a new heart, but his heart is not working properly. Now we put a pad to support this heart until he got the new transplant. So that is bridge to transplant. 1992, first in US to discharge a patient home on LVAD. So in 1992, we are the first center to discharge a patient who had an LVAD. So this is the patient. So you can see we are the pioneer here. 2000, the first in US to implant a Jarvik heart. In 2003, first in US to implant HeartMate 2 LVAD. 2012, first in US to implant continuous flow total artificial heart. So, and then first in Texas to implant mini syncardia total artificial heart. So what are the survival with these LVAD surgery? As you can see, the survival means that in due course of time, how many patients got survived. So in this Kaplan-Meier curve, you can see that our survival is pretty good. You can see the heart, this is HM2 means HeartMate 2, HeartMate 2, HeartMate 2. Bridge to transplantations, bridge to transplantation. DT means destination therapy. So this is not the statistics of our institution. This is the statistics of entire United States published in different journals since very early, 2001 to 2011. So this time frame. And these are the different pictures of the different patients. They already got the VAD implantations. And this is Dr. Bud Fraser, who do most of the VAD implant in our institutions. So the current generation of LVAD used most frequently at our institution. The first generations developed by Thorotec Corporations and approved in January 21, 2010 is the HeartMate 2. So this is the HeartMate 2. It's centrifugal. It's an axial flow pump centrifugal. And this is hardware approved in September 27, 2017. It's approved by, it's a product of Medtronics company, and the name is Hardware HVAD. So this is the HVAD, how we can see the blood goes. And this is the impeller, this is the housing, and how blood, impeller, housing, what is the way, how it moves through the device, through the motor. And the latest, most important, the new magnetically elevated technology. This is the HeartMate 3, approved October 18, 2018. And in our hospital, we mostly use this and this nowadays because these are the very important third generations, the LVAD, and that have a very high, nice results with in terms of outcomes. So Texas Heart Institute and Baylor experiencing with LVAD. As you can see that we are the more LVAD than any other program in the United States. So we do a lot of LVAD surgery in our institution, and that is that the Baylor St. Louis medical system historical VAD volume. If you see, you can see that how much we do every year, and it's increased exponentially. And if you look at the survival rate, 2012 71% to 2017 we have 90% survival rate. And here is the Intermax. Intermax is the all the data combined together throughout the US. So we are better than this. They have 84%, we have 90%. So we're doing pretty good here. So 5 decades later, the famed surgeon Bart Fraser continues quest for his artificial heart. So this is Dr. Bart Fraser, as I showed you, in 1968. These pictures, and then he still continues to develop the newer device, more biocompatible, less problem causing device. He's devoted his entire career. Now he is in 2009, he was 8 years old, now 2020. So I'm very much fortunate to work with him and collaborate with him. And this is, and I need to know a lot of things, learn a lot of the things regarding the device design, how it works, the device improvement technologies. He is the living legend. He is still working, and he has a plan. As he told me that I still need to finish this total artificial heart, and he told that he's not retiring. He still want to work for a couple of years. So as I told you that this is the picture, internal pictures of LVAD. So the blood comes here and goes through this. So as you can see, there are several hot spots because there is a pump. And it's a rotor, it's moving, and the blood goes through this, and blood is continuously experiencing high level of non-physiological shear stress. So shear stress, that means blood is in direct contact with the artificial biomaterial or experiencing high level of shear stress. So what happened? Because blood is, so if you look at the biology, now I'm talking about a little bit of basic science. So basic science, if you see, if the blood continuously goes through this device, so use your common sense. What will happen? The blood can get damaged because the high shear stress, blood contacting biomedical device, and blood is circulating again and again and again and again. So the platelets can be dysfunctions, the red blood cells can cause damage, hemolysis. So a lot of things can happen. But with the newer technology, every day we try to improve this, how to mitigate these problems, and how to prevent the post-implant clinical complications in those patients who have this device. So that is my area of interest, because I am not a doctor, I am not an engineer, but I know that what component or how this blood can get damaged, and which platelet part can get said, and what are the molecular mechanism behind this, and how we can mitigate this by improving the newer VAD technology, which have the capability of more biocompatible. So here is the case. So I'm a basic science researcher, and I do work with the clinical surgeon, I do work with the engineers, because we all have to work together to come into the solution for the betterment of patient, because this is the case. This is the importance of basic science, and the basic science can answer the basic fundamental questions of the biology, and through that knowledge, the engineer can be educated and can have the power how to improve, can have the data from the basic science to apply into the field and to improve the design, and the clinicians or the surgeons. These three triad can work together, patient management. Now, as I told you that the heart transplant is the ultimate for heart failure. So if you have a heart failure, you need a heart. So either you got a transplant or go to the another pathway, that is the device. Is it easy? No. The first you need a donor. If you got a heart attack and you need a heart, you need to identify a donor. Okay, I can donate. No, you cannot. It has to be matched. Then what do you need? Then we need a transport system. Suppose I got a heart attack and I'm admitted to one of the hospital in Calcutta, and another patient may be a potential donor. He is in Mumbai, or he is in some distant Darjeeling or somewhere else. So how it can be available? So we need a proper transport systems. So if we have a transport system, is it easy? No, there is a time. live for longer duration. Once we get a donor heart, it has to be put in the ice, and then it has to be transported for transplantations. A very short time. It's not a 10-hour, 12-hour job. It's in India within 2 to 3 hours, or sometimes maximum 4 hours or 5 hours. So how the heart transplant procedure is? The heart transplant procedure, I'm not going to discuss the elaborate surgical procedure, but I'd like to mention in a very layman's language how it works, how it happens. So this is the donor heart. This is the recipient heart. So this patient heart is failing, so we need to remove this heart. We need to put it back here, and then we need to stitch all other parts, and you're done. This is so simple? No, this is not so simple. Our heart, we got it, and then we have to wait it. And the patient who extremely highly skilled surgeons effortlessly happen. So this is the donor heart. It need to be implanted in the recipient's body. So once the heart in a place, they need to stitch it back. They need to attach all the. Then they have to use the electric shock or defibrillator to start the heart pumping, so that the heart is easy. No, it's not easy, but this is the case. So Texas Heart Institutions